In 2009, the National Cancer Institute initiated 12 centres nationwide to investigate the application of physics to oncology in the hope of finding new ways to understand, cure and manage cancer. Cancer cells are often recognised by their irregular shape. They seem squishier than normal cells, but just how soft are they? At Arizona State University in Tempe, physicist Dr. Robert Ros is studying the mechanical properties of cancer cells and comparing them to normal cells. Entering his lab is like stepping into the Doctor Who TARDIS. A dark revolving door, like an airlock, minimises vibrations and ensures that no light enters the room from the outside. Students are busy fixing cells on slides for the next experiment and analysing data from the previous day. The most intriguing equipment is a big black box on a bench to the right. Inside is not just the optical microscope that Robert uses to view the cells he studies. Directly above it is an atomic force microscope. That can trace the shape of the surface of individual cells. Rory Staunton is a first-year graduate student in the lab.
Because this equipment is so delicate, we've surrounded it with this foam to damp vibrations, and we've also placed it on a table that damps them as well.
The atomic force microscope is ingeniously fitted with a cantilever with a point so tiny it can press into precise areas on a cell surface. The cantilever tip on the microscope is almost impossible to see, but fortunately Dr. Ros has a large-scale model.
In principle, it has a cantilever, and on the cantilever is a tiny tip, and this is all small, so that it's in the size of maybe a hair or something. And we're using this to indent or to press on cells and to measure the response, which gives us some hints about the mechanical properties of single cells.
The amount of force it takes is then measured to give an accurate value for the cell's elasticity. And precisely where the tip indents the surface can be seen and mapped using the optical microscope.
And the cool thing about it is that they're aligned. So when we scan the surface of something, the laser light and the tip will move in unison, so we know what's happening mechanically and optically at the same time. We can get three-dimensional images with this by taking consecutive images in different focal planes and then stacking them all together. And so what we can do is indent the cell and then get a three-dimensional image that actually can measure the deformation. So we can squeeze it, apply pressure, and look at the shape change.
But this model represents a cell fixed to a microscope slide, and cells stuck on a hard surface don't say much about the ones growing in a tumour surrounded by other cells. So the main thrust of Robert Ros's team is to make the experiment more biologically relevant by embedding the cancer cells in a type of gel that resembles the consistency of the extracellular matrix found in the human body. It's a challenging project, but it should yield some interesting data.
For example, we're going to look at the mechanical properties of both cancerous cells and regular cells. And a lot of research has so far suggested that the cancerous cells are more compliant. So we expect that when we probe the cancer cells, we'll actually be able to indent them further with the same force. And there are probably a lot of metabolic processes and genetic factors involved in the differences between the structures of the cancer cells and the normal cells. And this setup is going to give us the ability to examine with the confocal microscope optically the different proteins that may be related to those structural changes. And one of the things that I'm interested in looking at is actually possibility of having a feedback loop between mechanical stimulus of like high pressure, which is something that you would see in a tumor-like environment, maybe actually triggering differences in the proteins inside the cells that could potentially cause a regular cell to behave more malignantly inside a tumor-like environment.
Fresh ideas like this could eventually transform our understanding of the mechanisms underlying cancer and may eventually lead to entirely new ways of treating the disease.